We are now in the second week of this course. I understand that it's sometimes hard to get up and running. If you have not started any of the assignments yet, you should probably get on those as soon as possible. In the first week, our content focused on an introduction to microbiology and some of the historical figures that contributed to the science, as well as the structure and function of cells. Many students were already familiar with the structure of eukaryotic cells from previous coursework. However, in this course we study prokaryotic cells in a lot more detail. Some of the drawings I received as part of this first week's assignment were not quite detailed enough. When I grade these, I look for evidence that you know enough about the structure to be able to answer questions on the exam. Some of the topics that students struggle with are the differences between prokaryotic and eukaryotic flagella, the difference between the cell wall structure of gram-positive and gram-negative bacteria, and the different types of movement that bacteria can use. So be sure to study the terminology and spelling associated with these topics. You will also see that assignment differs a bit this week. Every week we will learn different content. Depending on the most important concepts that I have to teach, different types of assignments will be used. There will always be a quiz for each chapter we cover and one other type of assignment. This week, the assignment is a concept map that I converted into a quiz-type assignment. Note that although the quizzes and assignments are open book and on-timed, it is smart to study first and attempt the assignment after that. Did you know that if I dock points from an assignment, I typically leave a note in the comments explaining what you did wrong? You can view the comment from the gradebook. On the right-hand side of the assignment, there's an icon that indicates there is a comment. When you select that, the comment will appear underneath that assignment. This week, you will be studying microbial metabolism. Again, you may have come across this before, but the emphasis in this course is likely different than what you've learned in the past because we focus on microbes. One big difference is that we introduce anaerobic respiration and contrast this with aerobic respiration and fermentation. Many of my students are confused by this because in the past they've learned about aerobic respiration with oxygen and fermentation without oxygen, and they probably did not learn about anaerobic respiration except for maybe a little footnote. In our course, however, we will come across many bacteria and protozoa that use anaerobic respiration. No oxygen is required for this process at all, so in that sense it is similar to fermentation. In all other aspects, however, anaerobic respiration is like aerobic respiration, except for the very last step that uses oxygen in aerobic respiration and some other molecule in anaerobic respiration. Different bacteria may use different molecules for that last step, but sulfur is the most common one. As for lab, we are waiting, not so patiently, on the last shipments to arrive. Most of what we need is here, and this weekend I worked with other lab staff on packaging what we could. I will keep you up to date as I get more information. For this week, I will make some separate handouts and hopefully some videos to prepare you for when the supplies are here. So keep working at it, and be sure to post questions regarding course content in the extra credit forum. As always, for anything else, you may call or text me at 352-559-9909, or you can email me through Canvas. Have a great week.